Hey there! How does the SARS-CoV-2 molecular test work? And what's the science behind it? Okay, let's say you're sick and you have symptoms like fever, coughing, but you're not sure whether you have influenza or SARS-CoV-2 virus. So you go to a testing clinic and they'll perform a nasopharyngeal swab. This is the easiest area to access for respiratory viruses or other infectious diseases. These include SARS-CoV-2, influenza, rhinovirus, bacteria like Streptococcus pneumoniae, or even microbes that don't cause disease and just want to hang out. So after the swab is done, you have to isolate the genetic material, and then the magic happens. PCR. PCR stands for polymerase chain reaction. This is basically a process of DNA replication inside a test tube, in vitro. So what are the components of PCR? Well, first you need your DNA template or your isolated genetic material. Then you add Taq polymerase. This is an enzyme that's capable of replicating DNA molecules. However, this is not like any other polymerase. Since PCR has to occur in very high temperatures, other protein polymerases might degrade or not function. Taq polymerase, however, came from a bacteria known as Thermophilus aquaticus. This is a bacteria that exists in the hot springs of Yellowstone Park. It's able to survive these very extreme hot temperatures, perfect for PCR. So once we have Taq, we also add nucleotides, which are the building blocks for more DNA molecules. Then we have a buffer filled with cofactors. This is to provide the ideal environment for DNA replication to take place in this test tube. And then primers. Primers are essentially small strands of DNA that specifically recognize a sequence, but I'll get more into that later. Once all of that is in the test tube, then we could put it into the thermocycler, which is basically a machine that's going to fluctuate temperatures. So that's PCR. But what's really happening inside that test tube? Well, let's take a look. So right now we're inside the test tube at room temperature. We can see here the template DNA, the different nucleotides floating around, the primers, and Taq polymerase must be somewhere around there. So this is before we go into the thermocycler. Once inside the thermocycler, the first step is denaturing. These temperatures are about 98 degrees Celsius, hot enough for the two DNA strands to be separated into individual strands. After denaturing has taken place, the next step is annealing. So temperature goes down to about 50-60 degrees Celsius, and the primers now recognize specific sequences of these separated DNA strands. But wait, there's that word again, primers. Originally I said primers are DNA sequences, short DNA sequences that can recognize a specific part of the template DNA. But what really is the purpose of primers? Okay, let's put it into perspective. Once you've performed that nasopharyngeal swab, there's a lot of DNA there that we just isolated. But we're just interested in one specific sequence that'll indicate to us that that's SARS-CoV-2 in this case. So we designed primers that can specifically recognize a sequence of the SARS-CoV-2 genome, and then we can amplify that, replicate it with PCR, and then detect it. So that's the importance of primers. So now that we're done with the annealing step, we need to perform the amplifying step, the final step of PCR. We raise up the temperature up to 72 degrees Celsius, and now Taq polymerase is able to recognize the primers and start adding nucleotides, extending the newly synthesized DNA molecule. This process is repeated multiple times until we have millions and millions of copies of whatever genetic material we wanted to replicate and detect, which in this case is a signal for SARS-CoV-2. This technique is thanks to a professor known as Kary Mullis, who won the Nobel Prize in Chemistry in 1993 thanks to this very same technique he developed of PCR. But wait a second. I've been talking about DNA this whole time, and as some of you might know, the SARS-CoV-2 genome is RNA. So how does it work if we're using RNA instead of DNA? Well, the version of PCR we're using is an updated one known as Rtpcr, reverse transcription PCR. It's the same as the original PCR technology, but with one additional step at the very beginning known as reverse transcription. What's going to happen here is that the RNA molecule is going to be recognized by another enzyme known as reverse transcriptase. This enzyme is known to function in retroviruses like HIV, and it can turn RNA into DNA. So reverse transcriptase is going to recognize the RNA template, add DNA nucleotides to it, and generate a DNA RNA hybrid molecule. And then that DNA RNA hybrid molecule is going to be read through PCR, and well, you know the rest. But let's get back to you. You were sick, you went to a testing clinic, they performed Rtpcr, and lo and behold, you tested negative for SARS-CoV-2. Congratulations. Thank you for watching. I just wanted to let you know, if you have any ideas for future videos, please comment down below. I will be happy to make any videos regarding any topic about biology or chemistry and any other sciences. Thank you.